Good morning, everyone. Today we are going to discuss about mechanism of oxygen binding. So let's begin. So mechanism of oxygen binding. See, in oxyhemoglobin, the ion is in +2 oxidation state, and it is in low spin state and diamagnetic. It is just the right size to fit in the hole of porphyrin ring. See, the low spin state of Fe2 plus is in the right size to fit into the porphyrin ring. But in deoxyhemoglobin, the Fe2 plus is in high spin state. Okay, so you know that what is low spin and high spin? In high spin, the unpaired electrons in d orbitals will be in uh, not in paired manner, whereas in low spin compounds, the unpaired electrons in d orbital will be in paired manner. That's the difference. Okay, the pairing is actually done by ligands. So due to the influence of ligands, the pairing takes place. Whatever. So in deoxyhemoglobin, Fe2 plus is in high spin and it is paramagnetic. See, when a compound converts to high spin from low spin state, its size increases. That's the thing. So the size of Fe2 plus increases by 28% when it changes from low spin to high spin state. Thus, Fe2 plus is too large to fit in the hole at the center of the porphyrin ring. So in oxyhemoglobin, the ion is in correct size to fit into the center of porphyrin ring, whereas in deoxyhemoglobin, due to its changes in spin, its size increases. Now it may not be fit into the center of porphyrin ring. So thus Fe2 plus is too large to fit in the hole at the center of porphyrin ring. Therefore, it is situated 0.7 to 0.8 angstrom above the ring, thus distorting the bonds around Fe. See, actually, in the previous session, we already discussed the ion is in octahedral manner in hemoglobin. But in deoxyhemoglobin, it changes. The shape changes to distorted octahedral. Okay. Then the presence of O2 molecule changes the electronic arrangement of Fe2 plus and also changes the complex from square pyramidal to octahedral. See, in this statement, in the third statement, we are dealing with the shape of hemoglobin before oxygen binding and after oxygen binding. Before oxygen binding, it is a 5-coordinated system. So obviously its shape will be square pyramidal, whereas when oxygen binds with the iron, it becomes octahedral. So oxyhemoglobin is octahedral, whereas the species that does not binds with oxygen, that doesn't mean deoxyhemoglobin, the species of hemoglobin before binding with oxygen is considered as a species of square pyramidal geometry. Okay, so during the binding of oxygen, there is a changes in shape. The lone pairs shifted to its shape. That's why the shape changes from square pyramidal to octahedral. This movement of iron atom in one subunit of hemoglobin acts as a kind of trigger which causes extensive structural changes in other heme units. This in turn influences the oxygen binding capacity of heme in other subunits. This is termed as cooperativity. It's actually cooperativity, not cooperativity. So it is cooperativity. Okay, keep in mind. So, what is it actually? When oxygen binds with the iron, its geometry changes. Its geometry changes from square pyramidal to octahedral. Okay, so when one of the heme unit changes its shape, it triggers the other heme units also to change its shape from square pyramidal to octahedral. So as a result, when one heme unit binds with the oxygen, all of them will definitely, as a result, will bind with the oxygen. since the first binds will trigger the other heme units to combines with the oxygen, and this is called cooperativity. An important parameter that affects the oxygen binding capacity of hemoglobin is the pH of the acidic medium. It's actually the pH of the acidic medium. The equilibrium between deoxyhemoglobin and oxyhemoglobin is shifted in favor of the deoxygenation process. Deoxygenation process, and this is called a Bose effect. See, the dependence of formation of oxyhemoglobin and deoxyhemoglobin is mainly based on the Le Chatelier's principle. So wherever there is a very good pressure of oxygen, definitely oxyhemoglobin will be produced, and if there is a very low pressure of oxygen, then deoxyhemoglobin will be formed. See, there is an equilibrium between these oxyhemoglobin and deoxyhemoglobin, and the medium. The pH of the medium depends which one will be produced, whether oxyhemoglobin or deoxyhemoglobin. It is determined by the pH of the medium, and it is called Bose condition. Bose effect. So in oxyhemoglobin, the oxygen is bounded to the hemoglobin in a bend end fashion. See, we know that in oxyhemoglobin, there is octahedral geometry, and the sixth position is occupied by oxygen in a band fashion. So FeO, there is a bond to another oxygen since O2 is present. So FeOO bonding is there. Then there is a bond angle since they are aligned in a bend fashion. Then those bond angle is 130 degree. So hemoglobin can interact with varieties of ligands such as CO carbonyl groups, cyano groups, etc. And we know that hemoglobin has very good affinity when compared to oxygen when they interacts with the carbon monoxide or cyano groups. That's why these gases like carbon monoxide, cyanides suffocates persons. Okay, this is how the carbon monoxide poisoning and cyanide poisoning takes place. So the bond of carbon monoxide with hemoglobin is so strong. So carbon monoxide never leaves hemoglobin. That prevents the oxygen supplies to cells. As a result, finally, the cells will die slowly. Then these molecules are toxic because they attach to the Fe atom of hemoglobin more strongly than oxygen, preventing the transfer of oxygen. Keep in mind, this may lead to death. Cn minus ions also interfere with the cytochrome enzyme system. This is the reason for the lethal activity. Okay, this is how the poisoning of carbon monoxide and cyanides takes place. And transport of oxygen and carbon dioxide. This is very important for your exam point of view. This is very important. This is one of the very frequently asking question. So hemoglobin has high affinity for O2 at high oxygen pressure. So whenever there is a very high pressure of oxygen, then hemoglobin definitely will binds with the oxygen. In lungs, the oxygen pressure is high. So hemoglobin can easily combine with the oxygen to form oxyhemoglobin, represented as HbO2. Then oxyhemoglobin is pumped into different parts of the body through blood. At muscle tissues, the oxygen pressure will be low, thereby oxygen will be released from oxyhemoglobin. At this time, myoglobin will take up the oxygen and will be stored as oxymyoglobin in tissues. So hemoglobin will transport oxygen as oxyhemoglobin, and myoglobin will store this oxygen and by forming oxymyoglobin. So after the removal of oxygen from hemoglobin, it takes up water molecules. See, we may think that after the removal of oxygen, the hemoglobin may directly interacts with the carbon dioxide, but it actually not like that. It takes up water molecules. So keep in mind, we have already discussed this topic. So at the muscle sites or at the tissue sites, water is taken up by hemoglobin, not carbon dioxide directly. The carbon dioxide released from the tissue combines with the water on the hemoglobin and produces bicarbonate ions and H plus ions. See, the carbon dioxide is not directly taken up by hemoglobin. Instead, water is taken up and the released carbon dioxide is dissolved in water and form H plus ions and bicarbonate ions. The H plus ions formed combines with the hemoglobin to form hemoglobinic acid. Hemoglobinic acid. Okay. And this is the reaction. Hb is hemoglobin combines with the water. Then carbon dioxide again reacts with this compound forming hemoglobinic acid and bicarbonate ion. So they sticks together. Okay, they never stay separated. The hemoglobinic acid sticks with the bicarbonate ion. Since bicarbonate is a base, hemoglobinic acid is an acid, so they can readily sticks with each other. Now the bicarbonate ions diffuse to blood plasma from hemoglobin. See, now the bicarbonate ions diffuse to blood plasma from hemoglobin. The blood returns to the heart through veins. It is then pumped to lungs and the bicarbonate ions converts back to carbon dioxide and water. See, at lungs, what happens is the reaction reverses due to the very low concentration of carbon dioxide at the lung site. The reaction reverses. The reaction reverses means hemoglobin become free and water and carbon dioxide will be released. This is carbon dioxide. Okay, this is CO2. There is CO. There is a wrong thing. CO3 is there. It's actually CO2. Correct it. Okay. And this is how the oxygen carbon dioxide cycle. That's all about today. And the next section is photosynthesis that will be discussed later.